Okay, good afternoon everyone. Welcome to lecture 36 of BMS 300. Today we're going to do the lymphatic system, and also we'll start our discussion on the immune system. But before we get started, I have an announcement. So if you look at the lecture learning objectives, which was uploaded previously, I have a new version uploaded now, and the main difference is that in that version the respiratory system was supposed to follow after the cardiovascular system, but now I moved the respiratory system to after the immune system, and that's what we'll get to next week, right after the exam. So please download that most recent version, and let's begin with an overview of what I'll go through today. So yesterday we were talking about the erythrocytes, or the red blood cells, and also we looked at the various type of white blood cells. Today we're going to specifically see how the various white blood cells are going to be produced, and we'll see the regulation of their production as well. And so in that regard we're going to go through this flowchart here, and it may seem complicated. There are only a few things which I want you to take away from this flowchart. Alright, so then we'll talk about the functions of blood. As you can see in this picture here, most of it you're already familiar with because we talked about the functions of blood in the integumentary system. Then we'll switch gears and talk about the lymphatic system, and we'll see what are the various components of the lymphatic system, namely the primary lymphoid organs, which includes the thymus and the bone marrow, and also secondary lymphoid organs such as lymph nodes, the spleen, mucosa associated lymphatic tissue, and so on. Alright, so with that, let's begin with the regulation of the production of white blood cells. So in this figure we see that regardless of whether it is all of the white blood cells we discussed yesterday using the abbreviation never let monkeys eat bananas, right? So whether it's all of those cells, or the blood platelets, or even the red blood cells, all of these are made in the bone marrow. They're all made in the red bone marrow from hematopoietic stem cells. So all of the cells and the formed elements in the blood, they're going to be made in the red bone marrow from hematopoietic stem cells. And so the question is, what causes all of these cells to be made from one type of cell? And the answer is hormones, which are called as colony stimulating factor, right? So CSF stands for colony stimulating factors. These are hormones which can regulate various white blood cell and red blood cell production. And so we find that these hematopoietic stem cells, they can differentiate into two lineages. You have the lymphoid stem cells and also the myeloid stem cells. What causes the hematopoietic stem cells to differentiate into lymphoid or myeloid stem cells is beyond the scope of our discussion, right? What we'll mainly look at is what happens to these myeloid stem cells further downstream to form either various white blood cells or red blood cells. And so these lymphoid stem cells will eventually turn into lymphocytes, as shown here. But notice that these myeloid stem cells, they are going to be acted upon by a CSF called the multi-CSF, right? So you have these multi-CSF, which is going to accelerate the production of granulocytes, monocytes, platelets, and red blood cells. So here are the red blood cells, here are blood platelets, and monocytes is a type of white blood cell. And the question is, what do we mean by granulocytes? So 3 among the 5 white blood cells, they are called as granulocytes because in the early years when they were being looked at by scientists, it looked like they had a number of granules in them, right? So that's why they're called granulocytes. Now we know that those granules are actually vesicles which are filled with various components which allow those white blood cells to perform their functions. In the case of neutrophils, it could be bactericidal compounds and other lysosomal enzymes, right? In the case of basophils, those vesicles can have components which can cause the inflammatory response. So all of those vesicles have different functions. So multi-CSF is responsible for the synthesis of granulocytes, monocytes, platelets, and red blood cells. And then we find that the erythropoietin is a hormone we discussed yesterday. that's going to lead to the formation of the red blood cells. I don't want to add any more detail than what I gave you in yesterday's lecture for erythropoiesis. Notice here that these myeloid stem cells can also form these really large cells called megakaryocytes, and these megakaryocytes, they have parts of their cell body just pinching off, right? And so these platelets will contain clotting factors. Platelets contain clotting factors, and that's what causes the blood clot formation to happen. Okay, now moving on, we find that there are these GM-CSF which can lead to the formation of both granulocytes and monocytes, right? So GM-CSF leads to the formation of granulocytes and monocytes. Now G stands for granulocyte. What does M stand for? What does M stand for? Monocyte. That's the most obvious answer, right? But actually it stands for macrophage, and that's the reason why I asked this question. It was too obvious. So monocytes are what eventually differentiate into macrophages, and so that's why M stands for macrophage and not monocyte. But yes, it is the GM-CSF which leads to the formation of both monocytes and granulocytes. Okay, and then these progenitor cells will be acted on by GM-CSF, which in turn can form these blast cells, and blast cells can in turn be acted upon by M-CSF, which stands for macrophage colony stimulating factor, which will cause the formation of monocytes, which can eventually turn into macrophages. You also have the you have the G-CSF, which leads to the formation of these various granulocytes. And the take-home message from this entire slide is what type of colony stimulating factor is responsible for the formation of various white blood cells. I'll repeat, what type of colony stimulating factor is responsible for the different types of white blood cells. That is the take-home message which I will hold you responsible for. And to highlight that take-home message, here is a poll everywhere question. Blank triggers the synthesis of eosinophils, monocytes, platelets, and red blood cells. So is it GM-CSF, multi-CSF, G-CSF, or M-CSF? You can turn to your neighbors or your friendly TAs and they can help you find the answer. Okay, let's bring it back together, folks. We have a lot of responses. Justin, anyone in your group has a response for us? Multi-CSF. All right, so most of you did pick multi-CSF. That is correct. And the reason is multi-CSF is going to help in the formation of pretty much everything except the lymphocytes, right? So that's why multi-CSF is the right answer. If I were to change the names of these white blood cells, the answer will change accordingly. Would you be able to answer it, right? That's the question for all of you. Did I hear no? I hope not. I hope you're able to answer it even if I change the names of these white blood cells. Yes? All right. Okay. Cool. All right. Okay, and here's the next question. Blank are white blood cells which are bilobed, which stain with an acidic dye, and they are involved in dealing with parasites and allergic reactions. So this is a question testing you on whether you can identify these 5 types of white blood cells based on the given description, and if you can at least mention 1 or 2 of their functions. So that's what this question is testing you on. You can again turn to your neighbors or your TAs and answer this question. Okay, we have a lot of responses. Let's see. Nell, anyone in your group has an answer for us? Eosinophils. Right. Pretty straightforward. I see that some of you did pick other responses as well. So we see that the only white blood cell which has a bilobed nucleus is the eosinophil. Kind of looks like a dumbbell. This is called eosinophil because it picks up eosin, the acidic dye, and it's also going to deal with allergic reactions and parasitic infections as well. Okay, so with that we move on now to the functions of blood, and much of what I tell you in this slide is a review of what we've discussed previously, but there are a few new things I'd like to point out as well. So the first function is to transport dissolved gases, nutrients, hormones, and metabolic waste, and we'll talk about how gas exchange happens in the respiratory system. We'll talk about how nutrients enter the bloodstream in the digestive system, and also how metabolic wastes can be removed is something we'll discuss in the urinary system. We already talked about hormones in the endocrine system. The next function is that blood can also help regulate the pH and the ion composition of interstitial fluids. And to explain how the pH is regulated, you have this reaction which is catalyzed by carbonic anhydrase, right? And this is a reaction we'll talk about, I think, 8 different times in a single lecture in the respiratory system, and so for now I don't want to spend any more time on this, right? But just know that blood can help maintain the pH, and also it influences the composition of the interstitial fluid. Moving on, we find that it can also help restrict the loss of fluid at the site of injury, and this was explained when we talked about the various steps in the wound healing cascade. So the very first step is hemostasis, where you have the blood platelets causing the blood clot formation, right? And so that is how we find that the blood can even prevent the loss of fluids at the site of injury. We also find that it can stabilize body temperature by redistributing heat which is generated by the muscles. And to elaborate on this point, I showed you this picture in the integumentary system. So if the body is trying to retain the heat on a cold day, we find that the vessels which are closer to the surface of the body will constrict, whereas the vessels close to the core of the body will dilate, and so the blood flow will be closer to the body core, thereby preventing the loss of heat. In contrast, if it's a very hot day, then the vessels which are close to the surface of the body will dilate, and that will allow the heat to be radiated to the environment on a hot day. So this is how the blood can help maintain the body temperature as well. The final function is that it can help defend against various toxins and pathogens, and this last point about defending against pathogens is what we'll discuss in the rest of today's lecture and in tomorrow's lecture as well. Right, so these are the various functions of blood. And are there any questions for me on any of these functions? Okay, if there are no questions, you know the drill. I have a question for you. The red blood cells or erythrocytes facilitate in the transport of gases by blank binding to the blank of hemoglobin proteins. So what gases bind to which part of the hemoglobin? You can turn to your neighbors and discuss this, or your TAs. All right, let's bring it back together, folks. Kamalani, anyone in your group has a response for us? Anybody in Kamalani's group? Yes. A. You picked option A, oxygen binding alpha and beta chains. All right. So when you look at most of the responses, most people said option C, where it says oxygen binds to the heme group. And is that right? Yes. Oxygen binding to the heme group is correct, because you have the iron in the middle of the heme group to which oxygen can bind reversibly. I noticed that some of you also did pick option D, where carbon dioxide binds to the alpha and beta chains, and that is correct as well. So we find that carbon dioxide can be transported by binding to the alpha and beta chains of hemoglobin, but oxygen also binds to the iron in the heme group. So when we get to the respiratory system, I'll tell you what are the other mechanisms by which carbon dioxide can be transported as well. Is this clear, folks? Yes? Can we move on? All right. So now we're going to switch gears and talk about the immune system and contrast it with the lymphatic system. So the immune system is referring to all the cells in the body and the various tissues which are going to play a role in immunity. And what we mean by immunity is the ability to resist infections and certain diseases. So when you look at the term, the words all the cells in the body and the various tissues, we see that there are different organ systems, and in these various organ systems they can play a role in the immune function. So we see that there are the parts of the lymphatic system which will play a role in the immune function. This is something we'll discuss later in today's lecture. The integumentary system also plays a role in the immune function. Just to remind you, I told you about the acid mantle which is on the surface of the skin that has antimicrobial activity. You have proteins like defensins which are also produced, which have this antimicrobial activity. So these are examples of how the integumentary system plays a role in protecting the body from pathogens. As far as the skeletal system is concerned, it is in the red bone marrow where you have the formation of both red blood cells and white blood cells, which play a role in the immune function. The cardiovascular system is going to pump blood, thereby moving these immune cells from one part of the body to the other. And the respiratory system and the digestive system is going to encounter pathogens based on what you're breathing in, and the digestive system will encounter pathogens based on what you eat, right? So there are these lymphatic tissues found in the respiratory and digestive system which can also deal with pathogens. So it is all of these systems working together which form the immune system. Now moving on to the lymphatic system, this is also called the lymphoid system. This too is made up of various cells and tissues and various organs which play a role in protecting the body, not only from environmental hazards but also from internal threats. So examples of environmental hazards could be the various pathogens which are in the environment. Internal hazards could include cells which, if they are abnormal, they can start affecting the body's own cells, right? So you have this autoimmune diseases. Even from that, the lymphatic system can protect us, right? So we'll look at specific examples of this later in today's lecture. But for now, the question is, what are the various components of the lymphatic system? So here's a cartoon showing you the various lymphatic vessels here shown in green. In the real body, it's not in green color, it's just colored this way for the ease of visualization. The lymphatic vessels, they're going to carry the lymph from the peripheral tissues all the way back to the veins, which will carry that back to the heart, right? So the next question is, what do we mean by lymph? So lymph is the fluid similar to blood plasma, but it is got none of those proteins which are found in the blood plasma. So in other words, it's the filtrate of the blood plasma. That's what lymph is. So lymph is a filtrate of the blood plasma. How exactly is it formed? We'll discuss that in the upcoming slides. So here are some lymphatic vessels in the lower part of the body. Here are other lymphatic vessels in the upper part of the body, and they can eventually carry the lymph through lymph nodes, which are also found in different parts of the body, like in the armpit or the axillary region. Now there are these two vessels I want to point out to you. You have this thoracic duct and the right lymphatic duct. And the reason I point out these two vessels is that it is these two vessels which will carry the lymph back to the cardiovascular system. So you see that the lymph from the left side of the body is brought by this vessel, the lymph from the right side of the body is brought by the other vessel, right? And so it's going to drain into veins called the subclavicle veins, which are below the clavicle. And from that, we move on to talking about what are the various primary and secondary lymphoid organs. So the primary lymphoid organs would refer to those tissues where the lymphocytes, they are going to be formed and mature, right? So primary lymphoid organs are where the lymphocytes will be formed and where they're going to mature. And this includes the various red bone marrow and also the thymus. So see, these are the two primary lymphoid organs. And in yesterday's lecture I told you about B lymphocytes and T lymphocytes. B lymphocytes are formed in the bone marrow, whereas the T lymphocytes, they are formed in the thymus. So now we move on to the secondary lymphoid tissues. These are tissues where the lymphocytes are going to be activated, where they can perform their function in dealing with pathogens. I'll repeat, the secondary lymphoid tissues are where the lymphocytes are going to be activated. So I want to use the analogy of a credit card which comes in the mail. Unless it's activated with your authorization code, which is usually the last 4 digits of your social security number, that card is pretty useless, right? So just like that, these lymphocytes, if they are not activated, they really can't perform their function. And what we mean by activation is that some of these lymphocytes are going to recognize pathogens and kill them, right? Whereas the other lymphocytes, they are going to produce proteins or antibodies which are going to target specific pathogens. And so that cannot happen unless these lymphocytes are exposed to the pathogens, and that exposure and activation of these lymphocytes is going to take place in the secondary lymphoid organs. Are we clear with that point? Yes? All right. So let us look at some examples of these secondary lymphoid organs. So you have the tonsils is an example of a secondary lymphoid organ. Also you have the spleen, where there can be activation, and the appendix, which is linked to the large intestine, and also all of these mucosa associated lymphatic tissues which are found in various parts of the body, and the lymph nodes as well. So let me just tell you a little more about these mucosa associated lymphoid tissues, abbreviated as MALT. So you're going to find these tissues in the respiratory system. Why is that? Because you could breathe in some of these pathogens, so these tissues will deal with those pathogens. You have it in your digestive tract because you could ingest certain pathogens which need to be dealt with, or you could also have pathogens exposed during sexual activity. That's why you have it in the reproductive tracts and in the urinary system as well. So wherever there's a chance for the body to encounter these pathogens from the external environment, you have this mucosa associated lymphoid tissues which are present to deal with the pathogens and train the body in the future to deal with such pathogens as well. Okay, is it clear why we need secondary lymphoid tissues? Okay, all right. Now in this figure on the left hand side, you see all of the various lymph nodes and lymphatic vessels are shown on the left hand side, whereas on the right hand side you see the different primary and the secondary lymphoid organs are shown in the right hand side. Now when we mention the word lymphoid cells, I only briefly talked about the T cells and B cells, but there are also these phagocytic cells and other cells which I haven't mentioned, right? We're just looking at a few of these cells in the lymphatic system. So this is an important slide where I want you to know the difference between primary and secondary lymphoid tissues, and so to make sure you understand the slide well, here is a poll everywhere question. So if you want to study how lymphocytes are formed and how they mature, then you would most likely harvest cells from blank. Would you harvest it from the lymph node, or from the tonsils, from the thymus, or from the spleen? You can turn to your neighbors or your TAs and they can help you answer this. Okay, Gabby, anyone in your group has a response for us? C for thymus. Okay, most of you did pick thymus. And so this question is asking about the primary lymphoid organs, because that's where you have the lymphocytes being formed, and that's where they are maturing, right? So it's in the primary lymphoid organs where they're going to mature and be formed. And there are two types of primary lymphoid organs we talked about. The first one is thymus. That's a given option here. What is the second example of a lymphoid organ? Bone marrow, right? So if bone marrow was given here, that's also a viable option. Okay, so now let's move on and talk about how lymph is formed. So this is a picture we went through in the lecture on connective tissue. Okay, we talked about this a long time ago. I just want to refresh your memory on what we discussed, and then I want to add a few more details to what was previously covered. So we see that this is a very simplistic representation of the cardiovascular system and also the lymphatic system as well. So here's the heart pumping oxygenated blood from the left side into these arteries, and this blood is being pumped at a very high pressure. And so because of this high blood pressure in these vessels, we see that some of the blood plasma is going to move into the interstitial space. The interstitial space is the space surrounding cells and also the blood vessels, and only the fluid will move into this interstitial space because the proteins are too large to move through the pores in the blood vessels. Okay, and this interstitial fluid is mostly going to be drawn back into the blood vessels because of the osmotic pressure exerted by the plasma proteins. So the plasma proteins stay behind in the blood vessel, exert an osmotic pressure, and they draw back the fluid back into the bloodstream. But we see that some of this interstitial fluid is going to, rather than going back into the bloodstream, it can move into the lymphatic vessels, and the moment it enters into the lymphatic vessels, it's called lymph. So when this fluid is inside the blood vessels, it's called blood plasma. When it's in the interstitial space, it's called interstitial fluid. When it's in the lymphatic vessels, it's called lymph. All right, and so we find that this lymph is not going to be moved through the body by the active pumping mechanism like how blood is moved, but just like how the veins move the blood back to the heart, we find that the movements of the body and also muscular contraction is going to help move the lymph back to the vessels, which will eventually take it back to the heart. Okay, so this is something we discussed previously, but now I'm going to give you a little more detail to explain why the interstitial fluid will actually enter into the lymphatic vessels. All right, we're going to see why this interstitial fluid enters into the lymphatic vessels. So in this figure here on the left, this is just showing you the direction of the flow of blood. So here's the arteriole through which blood is flowing in this direction, and some of this blood flows down into the capillaries as shown by these arrows, and from the capillaries it goes back into the venule to be oxygenated in the lungs. But some of this fluid is going to move from the blood vessels into this interstitial space, and from the interstitial space it goes into the lymphatic vessels, and the question is why. And we find that the reason is that these lymphatic vessels, they have these endothelial cells which are bound together loosely. Right? These lymphatic vessels are made up of endothelial cells, made up of these cells which are bound together loosely, and they're going to form this one-way valve, as you can see right here. So they're going to overlap with one another, thereby forming a one-way valve. And so when the pressure is high enough, it's going to slip in through this one-way valve, but then the fluid cannot get back into this interstitial space. So the fluid can only flow in one direction. Along with this interstitial fluid, we also find that pathogens like bacteria and viruses can also get into this lymphatic circulation. Once it gets in there, it can't escape. And it kind of reminded me of this trap here, right? So here's this mouse walking down the trap, boom, it goes in, and it can't escape out, right? So similarly, because of these one-way valves in the lymphatic vessels, the pathogens can't escape back to the interstitial space. Is that clear, folks? How many of you have dealt with a mouse in your house or room? Right? Especially dorms can be notorious for that. We can talk about it later, but I hope you can appreciate this trap, which is not hurting the mouse in any way, right, for now. Okay, moving on, let's talk about the primary lymphoid organs, and the first one is the thymus. So the thymus is going to be found above the heart, superior to the heart, and in children it's quite large, but as we start developing and during puberty it's going to start atrophying, and so that's why it's understandable why children can have a greater immunity when compared to older individuals where the thymus is already atrophied, and why that is you'll be able to appreciate as we go through this slide. And so the T cell development and maturation is going to happen in the thymus as we discussed previously. So a cross-sectional view of the thymus indicates the outer part, which is the cortex. What do you think the middle inner part is called? If the outer part is called cortex, what is the inner part called? Starts with the letter M. Come on guys, yes. What is it? Medulla, very good. So the inner part is called the medulla. So in the cortex you have the T cells dividing and they're multiplying, and then they're going to move into the inner region, the medulla, where they will actually mature, right? And so this maturation process, we're going to talk about it in the next few steps, but once they mature, they're going to leave the thymus through the various blood vessels. Okay, now let's talk about this maturation process, which is taking place in these thymic epithelial cells. So these are specialized epithelial cells found in the thymus, and this selection is a two-step process, right? The first step is where selection is done for efficacy. The question is asked, are these cells able to recognize proteins? Yes, if they can recognize proteins, they can move to the second step, where a selection is made for specificity. Now in the second step, if the cells are found to identify the body's own proteins, then that's a problem, and those cells have to be terminated, right? So if I were to use anthropomorphic examples, as a former mentor mentioned this, it's like there's this handshake, hello, how are you? They come to these thymic epithelial cells and they interact, and the first question is, are you able to recognize proteins? And if the answer is yes, then this T cell can go to the next level. And in the first step itself, if this T cell cannot recognize proteins, then this thymic epithelial cells will tell it, please go die, right? There's no point in you surviving. And what it's going to do is it's going to withhold growth factors, and as a result, this cell will die. But if it goes on to the next step, the second question is, can you recognize the body's own proteins? And if the answer is yes, then that cell should be destroyed as well, because now this cell which can recognize the body's own proteins is an internal threat, and that has to be dealt with by the lymphatic system. Is that clear? And so that it should be able to recognize proteins which are not the body's own proteins, and that's when these T cells can be allowed to leave the thymus. And this is a very stringent selection process. Around 1% of the total T cells are finally selected. Okay. So now let's look at a few examples of the secondary lymphoid tissues and where they are found. Notice that you have these tonsils in the pharynx, which can deal with pathogens which enter through the nasal passageway. You also have tonsils in surrounding the oral cavity, the palatine and the lingual tonsils, which can deal with pathogens that enter through the oral cavity. And even in the digestive tract, we have these in the intestines, you have these crypts, which are like deep pits, and inside that, if the pathogens come in, they can get trapped in this lymphoid tissue and they can be destroyed, and the fragments from those pathogens can be used to train the immune system to deal with future pathogens which match those fragments. Right, and we find that the digestive system can be considered like a donut, right, where the one hole is in the mouth and the other one is in the anus, And so our digestive system is like a really long tube which is exposed to the external environment, right? And so that's why you have all of these lymphoid tissues found in the digestive tract to deal with pathogens which enter into the digestive system. Okay, any questions, folks? We'll talk about these lymphoid tissues when we get to the digestive system as well. Now here's an example of another secondary lymphoid organ. These are the lymph nodes, and we will just see what are the various components and the exact process of how the lymphocytes are activated is something we'll get to in tomorrow's lecture. But for now, notice that there are these number of afferent vessels which are bringing lymph to the lymph node, but you have a single efferent vessel. And so what this means is that the lymph is going to spend a longer time inside the lymph node because it has multiple paths of entry but only one exit pathway. So the lymph can spend more time in the lymph node, and consequently if any activation needs to take place, pathogens need to be dealt with, that can happen in the lymph node. There are also all of these blood vessels which bring oxygenated blood and carry away deoxygenated blood as well. And the outer part of the lymph node is known as the cortex, and the question is, what is the inner part called? Medulla, right? That's also called the medulla. Now in the cortex you have these structures called follicles, and in the follicles the outer part has got naive B cells. So these are B lymphocytes which have not been exposed to pathogens, but then in the middle of this follicle you have a germinal center where these lymphocytes which have been exposed to pathogens are undergoing proliferation. That's what they mean by clonal expansion. Exactly what is happening is something we'll discuss in tomorrow's lecture, right? How this clonal expansion takes place. There are also some of these T cells which are going to be in transit, and they'll have a surveillance role. Again, what do we mean by the surveillance role? Again, that's something we'll discuss tomorrow. Now moving down to the medulla, you have these plasma B cells which can produce antibodies. So these are cells which have been activated to deal with pathogens. They can produce antibodies for specific pathogens. And you have also macrophages which can eat up pathogens if they encounter them. And there's this middle region called the paracortex, which is between the medulla and the cortex, where you have these dendritic T cells which can also deal with pathogens as well. So having this described for you the various components and the different kinds of cells found in the lymph node, let's just consider the overall function of the lymph nodes. So the first function is to clean out the lymph of any pathogens which may be encountered. So these dendritic cells, the macrophages, all of those can eat up the pathogens which are encountered and destroy them. And the fragments from those pathogens can be used to activate the immune system, right? And so these dendritic cells and macrophages are called antigen presenting cells. And so as shown in this cartoon here, they can present the pathogens which they have destroyed to the naive T cells, right? And once it has been presented, let's say with this fragment, which is the hat worn by this pathogen, now this activated cell can detect other cells which have that type of protein, and it can kill it using this tiny sword here, right? And the sword is referring to proteins which can cause holes to appear in the cells which it is trying to destroy. Right, so this is just a high-level view of what's happening in the lymph nodes, but we'll get into the details of this in tomorrow's lecture. Here's another example of a secondary lymphoid organ. The spleen also does a similar function. So the spleen is going to be found towards the lateral side of the stomach, and a cross-sectional view indicates that it's got a red pulp on the outside where there are many red blood cells, and it's got the white pulp on the inside which has more lymphoid nodules. In yesterday's lecture we talked about how the red blood cells, they are going to be monitored by macrophages in the red pulp, and so the abnormal red blood cells will be phagocytosed, and we find that the iron from those red blood cells can be temporarily stored in the spleen as well. That's another function for the spleen. And in this white pulp we find that the innate immune response the immune response can be initiated when pathogens are encountered. Just like how we saw in the lymph node, similarly, an immune response can be initiated when pathogens are encountered and the various lymphocytes can be activated. So I'd like to quickly summarize the functions of the lymphatic system. We see that it's going to produce the lymph, it's going to maintain and distribute lymphocytes to the various lymphoid tissues. We also find that any of the excess fluid in the lymph is going to be, from the tissues is going to be turned back to the circulatory system. And also the composition of the interstitial fluid can be maintained by the lymphatic system as well, by dealing with hormones, nutrients, wastes. All of this can be sent back to the circulatory system. So this is the functions of the lymphatic system. So in tomorrow's lecture, we're going to talk about the two types of immunity, the innate versus adaptive immunity. Innate immunity is the immediate response. Adaptive immunity is the response after the body has been trained, right? So in order to talk about that, I need to give you a few definitions, right? So we'll talk about 4 different definitions and then we can end class. So the first definition are PAMPs, which stands for pathogen associated molecular patterns. And so an example of a PAMP is the lipopolysaccharides, which are found on the surface of gram-negative bacteria. So whenever the body detects these lipopolysaccharides, it's going to trigger an immune response because it signals that the presence of pathogens in the immune system. So this is the first definition. A second definition is DAMPs, which is referring to damage associated molecular patterns, and these are unusual molecules which are found in the extracellular fluid. So molecules which should not be found in the extracellular fluid, if they are present, it indicates that a cell has been damaged. An example of this is that if you have a high level of ATP in the extracellular fluid, that's an indication that a cell has been damaged, because we know that making ATP is pretty hard. There are a number of steps involved, and if you have high levels of ATP in the extracellular fluid, it indicates that some cell has been damaged, and as a result there will be a immune response as well. Now the third definition is the term CD, which stands for cluster of differentiation, and these are proteins which are found on the surface of cells, which have a twofold effect, right? So as far as we are concerned, it helps us in identifying a given cell if it's expressing a type of CD protein, but as far as the cells are concerned, it plays a role in cell-to-cell signaling. And to highlight this, I want to give you an example of the CD8 protein, how it is expressed on a surface of a cell. So here's the infected cell shown here in orange, and this infected cell is displaying a fragment of the pathogen which infected it on this MHC1 protein. So MHC1 protein is displaying a fragment of the pathogen which infected this orange cell, and this orange cell is basically saying, please destroy me because I've been infected and I have no hope of recovery. And this CD8 cell is expressing the CD8 protein on its surface, shown here in pink. Now the CD8 protein is going to bind to this MHC1 protein, and it brings the two cells in proximity to each other. It's kind of like one pirate ship throwing the grappling hooks on the merchant ships, and it brings the two ships in close proximity to each other. So once they get in close proximity, the CD8 cell is going to release perforins, which will cause holes to develop in this infected cell and thereby destroy it, right? So it is because of these perforins where I had the little cartoon of a tiny sword which can make holes in things. Similarly, the perforins can make holes in the infected cell and destroy it, right? So I hope you see how the CD8 protein can help in cellular signaling. Now the final definition I have for you is cytokines. I mentioned cytokines before, but these are proteins which are released by cells, and they can play a role in signaling, right? And so the signaling could be autocrine, meaning that the cell which released it to that very same cell, this cytokine can bind. We'll see an example of that. You also have paracrine signaling, where it's sent to a close by proximity to bind to other cells, or it could be sent to a long distance by having the cytokines sent in the bloodstream, through in an endocrine fashion. And so the purpose of these cytokines could be few. You could have growth factors sent, you can have interleukins, and you can have these chemokines which will induce chemotaxis. And what we mean by chemotaxis is that when a signal is sent out, other cells are going to show up at the desired region. So it's kind of like shooting out a flare, right? And then people know where you are and they can come and rescue you. Similarly, these cytokines can help in chemotaxis. And so we see that as a result of these cytokines, there can be cellular motility where cells arrive at a specific region. There could be differentiation of other cells. There could also be changes in gene expression. So all of these points here, I've previously mentioned it when I walked you through this figure. Remember this figure? We talked about it a long time ago. So first question, do you remember this figure? How many of you remember this figure? Okay, good. A few of you remember this figure. For the rest of you who didn't raise your hand, we talked about this figure when we discussed different types of cell junctions. Do you remember what cell junction we were talking about? What is it? There are three kinds of cell junctions. You have gap junctions, we have anchoring junctions, and tight junctions. Okay, good. Tight junctions, right? So among tight junctions, we talked about the Claudin proteins, which are pretty important. And so this figure was saying that in the digestive tract, if the Claudins are not functional, you can have pathogens coming in, and those pathogens are detected by these macrophages and dendritic cells, right? So these are cells which play a role in the innate immune response. Guess what? They are releasing cytokines, right? They are releasing cytokines, and as a consequence, it's going to activate these T cells, right? And these T cells, they're going to have a further effect on affecting Claudin expression. So you see how the cytokines are having an impact on affecting gene expression, and eventually it can also lead to problems like tumorigenesis if epidermal growth factors get in. But the key here is we're going to talk about cytokines. When we come back tomorrow, we'll talk about DAMs, PAMs, and the CD4 and CD8 proteins as well. All right, so with that, let's end class 2 minutes early, and I'll see you back tomorrow. All right, thank you for your time.